Pharma sir, you can start sharing. Sir, first of all, please unmute, sir. Pharma sir, you can start sharing. Sir, first of all, please unmute, sir. Pharma sir, you can start sharing. Sir, first of all, Pharma sir, you can sharing. Sir, if you are having technical problem, I will share the slides from here, sir. Sir, please unmute, sir. Sir, your screen is visible, but you can actually enlarge if possible. Please unmute, sir. So you're still in a muted condition. Please unmute. So you're the host now. You are the host now, sir. All the controls are with you. Just press the mic button. Mic button is there, so just press it and it will open. Sir, just unmute, sir. Just press the mic button. It will unmute and you can start presenting. Red color mic button is there, now, sir. Just press it.
Uh, so are you able to hear me, sir?
Yes, sir. We are able to hear you.
Okay, okay. Okay, I'll start now, sir.
Yes, sir.
So the applications of electrospinning. So when a high voltage is applied to a liquid droplet, the body of the liquid becomes charged and the electrostatic repulsion, it will counteract from the surface tension, and the droplet is stressed at a critical point. A stream of the liquid will erupt from the surface. This point of eruption is known as the Taylor cone. The elongation and the thilling of the liquid stream leads to the formation of the uniform electrospun nano from the structures. The nano from fibers with the diameters ranging from few nanometers to several from the microns can be produced using an electrostatically driven jet of the polymer solution or the polymer melt. The word nano is obtained from the Greek word nanos, which equates to dwarf or something extremely small. Technically, 1 nm is equal to 1 from the billionth of a from the Thus, all the nanoscale structures typically have a dimensional range or length between 1 and 100 from the nm. So this slide shows the diagram of the electrospinning apparatus. In this slide, you can see both the horizontal electrospinning apparatus and the vertical electrospinning apparatus. Mainly, the electrospinning equipment has 3 components. It has a capillary tube, or from the stainless steel, even the needle. It has high voltage from supplier, 2 to 50 kilovolts, and also it has a drum-like structure or a revolving screen. So in the horizontal electrospinning apparatus, it contains a syringe pump, and within this syringe you can take the liquid containing the drug plus excipient, and here we'll be having a stainless steel needle. We have to apply the high voltage, and when we apply the high voltage, we can obtain the electrospun nanostructures. This can be obtained on a revolving steel structure. Then again, if you take the vertical electrospinning apparatus, here we are having a high voltage from the power supply, and within the syringe, we have to take the organic solvent or aqueous solvent, in which you can take the drug plus the excipient, and then it can be sprayed onto a revolving drum-like structure. So here we can see the vertical electrospinning apparatus. So in the electrospinning, here, for example, so many from even the patents have been filed on the process of electrospinning. In 1969, Taylor published his work on the shape of the polymer droplet produced at the tip of the needle when an electric field was supplied. Taylor found that the pendant droplet developed into a cone when the surface tension was balanced by the electrostatic force, and the jets were ejected from the vertices of the cone with the diameters smaller than the diameter of the needle when the electrostatic force exceeded values of the surface tension. In the 1971, Baumgarten reported to make an apparatus to electrospin acrylic fibers with the diameters in the range of 500 to 1100 nm. In 81, Larondo and Manley successfully produced polyethylene and polypropylene electrospun nanofibers from the drug plus the excipient from the melts. The synthetic and the natural polymers have been used to prepare the electrospun nanofibers. So the electrospun nanofibers have been employed in the drug from the even the delivery applications. Electrospinning is a very simple, versatile, and efficient process by which we can prepare the polymeric nano from the fibers from the electrostatically driven jet of the polymer solution onto a substrate. In the field of drug delivery, electrospun nanofibers hold promise for the controlled release formulations. The submicron fibers produced by the electrospinning process. They have the advantages like tunable from this pores, high surface area to volume ratio, tailor the nanofibers with the desired composition in order to achieve this specific function. The advantages are they have very small pore size, high value of pores, and the large from the surface area. These from this porous non-woven polymeric networks, they have high value of the fiber from the even this connectivity. They can be employed to control the nanofiber composition to achieve the desired property or the functionality, offering more flexibility in the surface from the functionalities. Now, in the electrospinning process, what are the important points which can influence the efficiency of the process? What is the viscosity, how much excipient we are employing, what is the molecular weight of from the polymer, elasticity, surface tension, applied voltage. Then we can have the needle to collector from the distance, volume feed rate, needle from the diameter, temperature, humidity, atmospheric pressure. So the important factors which are affecting the electrospin from this nanofiber preparation are molecular weight of the polymer. The diameter of the fiber increases with the molecular weight. The low molecular weight polymers result in the formation of the beads. Viscosity, fiber diameter increases as the solution viscosity also increases. Solution concentration, fiber diameter decreases with the solution concentration. Surface tension, nanofibers without beads can be obtained by decreasing the surface tension of the solvent system. Voltage, average diameter of the nanofibers increases with the applied voltage. Flow rate, the diameter of the nanofibers decreases as the flow rate decreases. Distance from the needle tip to the metal from the collector. When the distance increases, average diameter of the nanofibers also decreases. Humidity, as the humidity increases, the interest from this connecting pores and the diameter of the fibers will decrease. Temperature, a temperature increase resulted in a decrease in the fiber diameter. Next, I'll tell about the natural from this polymers used in the electrospinning process. Chitosan, gelatin, silk, fibroin, these have been widely employed. Synthetic polymers are also employed in electrospinning like PVA, polyvinyl alcohol, polyepsilon caprolactone PCl, polylactic acid PLA, polyglycolic acid PGA, and even PLGA can be employed. Chitin is obtained from the crab and the shrimp. Chitin upon deacetylation gives the chitosan. So what are the polymers and the solvents used in the electrospinning process? Suppose if you're employing celloacetate as a polymer, we can employ solvent as a stone and the dimethyl estamide and ethanol in a specific ratio, 3:2:1. For the chitosan, we can employ the ultra pure water along with 0.7% acetic acid because this polymer dissolves only in the acidic pH. Ethyl cellulose, we can employ water plus ethanol plus acetic acid. Gelatin, we can employ acetic acid within the distilled water. Gum arabic or acacia, we can employ the deionized water. And for pectin, we can employ the water with 2% acetic acid. Polyethylene oxide, PO, we can employ the deionized water. Polylactide co-glycolide, PLGA, we can employ from the tetrahydro from this furan, penan, dimethyl formamide as a solvent. For PVP, you can employ anhydrous ethanol. For polylactic acid, PLA, we can employ chloroform and acetone as the solvent blend. Next, if you take from the chitosan, when from this nano formulation has been prepared by employing the anti-tumor drug doxorubicin, here from the ionic from the gelation technique was employed. Chitosan interacts with the polyanion dextran sulfate. It gives extended release of the anti-tumor drug doxorubicin. Chitosan nanofibers, they have been prepared by employing various excipients like the carboxymethyl from the chitosan, carboxyethyl, chitosan, hexanoyl, when the chitosan-based nanofibers have been successfully employed by electrospinning of the chitosan blended with the polyethylene oxide, PVA, silk fibron, and when with the polyepsilon from the caprolactone and even the agarose. Chitosan has been widely employed in the various drugs from the delivery applications. These have been employed in the tablets, capsules, microspheres, nanoparticles, films, and the gels. And the various formulations have been employed like the matrix coating, capsule shell, emulsion cross-linking, spray drying, ionic from the gelation, sieving technique, ionic gelation, solution casting, and cross-linking. And various drugs have been employed like the 5-amino from the salicylic acid, diclofenac sodium, theophylline, insulin, anti-infective drugs, anti-ulcer drugs, ascorbic acid, an anti-tumor drug, doxorubicin, and anti-infective drug, ofloxacin. So in the drug delivery applications, the nanofiber drug delivery systems, it has been employed with the various drugs, growth factors, and here the drug release occurs from the polymers, mainly by the two ways. One is erosion and the other is the diffusion process. Most from this polymers are degraded by the hydrolysis and the enzymatic decomposition. Even the anti-infective drugs have been loaded in the electrospin nanostructures. So here, what are the electrospin nanofibers we are taking from the Diller et al. They have prepared the nanofibers by taking the polylactic acid PLA and polyepsilon caprolactone single and the biocomponent from the polymers. So the drugs used are from the tetracycline, chlorotetracycline hydrochloride, amphotericin B. These have been evaluated for the release properties and the anti-infective activity. Solvents used were chloroform and THF, that is tetrahydrofuran, or toluene as an organic solvent for the polyepsilon caprolactone, polylactic acid, and for the biocomponent polymer, that is polyepsilon caprolactone and the polylactic acid electrospin nanofibers. So here from even the silk from this fibroin has been employed. Silk fibroin, it has been employed actually as a bio from this. It has the advantage that it can be decomposed after the complete drug release. And here the various electrospin nanofibers has been prepared by employing the hexafluoro 2 from this propanol as an organic solvent. Silk from this nanofibers can be employed as scaffolds for the tissue engineering. This can be employed in the wound healing. There's a wound dressing applications. So here how it all they are used from even the calf skin dissolved in the organic solvent, electrospin from the nano scaffolds were prepared mainly for the wound healing, wound dressings, and in the case of the tissue engineering applications. So here, suppose if you have the tumors in the area of esophagus, it can be treated. And for this, we have to employ the anti-tumor drug. On the stent, it can be placed in the area of the esophagus. So here we can take an anti-tumor drug from the paclitaxel, and here the excipient employed the polyepsilon from this caprolactone, This can be blended and this can be electrospun actually on a bare stent, and this can be placed in the esophagus so that we can treat the tumors in the area of the esophagus. Similarly, if you take the hydrophilic and lipid soluble, various types of drugs have been electrospun both for the oral applications, and here if you take from this vitamin from the B12, orally the absorption is poor because quite hydrophilic, and in this case the drug absorption can be enhanced from the GIT, and also it can be employed as a injectable route of administration and also as a transdermal patch. So the electrospun nanostructures can be employed as a transdermal patch. It has a lower toxicity. It is non-invasive. It can be easily applied as a transdermal patch. Vitamin B12 loaded electrospun nanostructures, they can be employed to absorb the drug across the skin, across stratum corneum into the blood. So here the electrospinning technique can also be employed for the vitamin A and vitamin E also. This can be employed as a transdermal patch by employing the celluloid. Celluloid can entrap the drug vitamin A and vitamin E in the transdermal patch. Then it will show from when this extend release, the drug release it can be extended up to 6 hours initially, and also complete 1 day we can extend the drug release. So it can be employed for the long-term absorption of the drug. So the electrospun nano from these fibers have been employed to entrap the various anti-inflammatory drugs. These anti-inflammatory drugs have been entrapped by taking various polymers like from the chitosan or from this PVA, polyepsilon caprolactone. The advantages are it gives a rapid onset of action, it has ease of approach, and it can avoid the extensive hepatic effect. Also, it can be employed, it can be placed under the tongue so that it can be employed for the sublingual administration of the drug. And we can achieve the extended release up to complete 1 day. Razia Begum et al., they have employed the electrospinning technique to enhance the dissolution rate of the hydrophobic drug, anti-inflammatory drug, by employing from this PVP, the drug polymer ratio is 1:4. We can enhance the dissolution rate of the water-insoluble drug, anti-inflammatory drug, by employing electrospinning technique. Zia Shen et al., they employed the Udragid L155 electrospun nanofibers for the diclofenac sodium. The drug is released at the end of the GIT, that is for the colon-specific applications. Initially in the stomach, very small amount of drug is released, but the entire drug is released in the area from the colon so that it can be employed to treat the inflammation. Electrospun diclofenac sodium loaded Udragid L155 nanofibers have been employed for the colon from the targeting of the drug. Simvastatin electrospun nanofibers were prepared for the oral extended release. The simvastatin electrospun nanofibers, it has been employed for the extended release of the drug, and the work has been published in the Journal of the Pharmaceutical Investigation, Springer Journal. Now the various steps of polymers and the drugs which are used for the immediate release formulation of doing electrospinning. Here we can take the examples of polyethylene oxide and the poloxamer for a drug like lovastatin, PVP and the soluplus for the anti-inflammatory drug, polylactic acid and butylene adipate, butylene from the terephthalate for the aceclofenac. Celluloid can also be employed for the alpha-arbutin. Hydroxypyl beta-cyclodextrin, it can be employed along with the other hydrophilic excipients like the PVP. This can be employed for the acyclovir, polyethylene oxide, and the poloxamer can be employed. Similarly, you can employ the PVP and ethyl cellulose for the anti-infective drug, that is ciprofloxacin, PLGA, and even this PVP can be employed for the anti-inflammatory drugs, moxifloxacin. PVP and the zine can be employed for the anti-inflammatory drug for this ketoprofen. Polymers and the drugs which are being employed for the extended release formulations. By doing electrospinning, they can be employed for extended release formulations. Example, the gelatin can be employed for the amphotericin B. PLGA can be employed for the ciprofloxacin hydrochloride. Polyvinyl alcohol and from this PVP can be employed for the anti-tumor drug. Similarly, other types of drugs like the ciprofloxacin also can be entrapped in the electrospin nanofibers for the oral extended release. So the other polymers and drugs employed for the extended release are PVP and PLGA. These can be employed for the moxifloxacin, polyethylene oxide. This can be employed for the anti-tumor drug from the doxorubicin. So the drug polymer here, in place of employing a single polymer, we can take a polymer blend along with the specific drug for doing electrospinning. Example, the anti-tumor drug, we can employ doxorubicin. Here we can take the polyepsilon caprolactone plus SLS. Similarly, we can take the anti-tumor drug from this along with from the PLGA, and some anti-infective drugs, they have been entrapped within the excipients like the PLA and the PEVA. Even other anti-infective drugs like from this, even the rifampicin has been entrapped. Similarly, we can employ SPAN 60 SLS, doxycycline, antibiotic drugs. PLG and the PG can be employed for the various anti-infective drugs. Polyepsilon caprolactone has been employed for the anti-infective drugs, and polyvinyl alcohol has been employed for the anti-infective drug. For anti-hypertensive drugs also, we can employ when the PLGA, and for anti-inflammatory drugs, we can employ it in these anti-inflammatory drugs can be formulated so the drug can be released towards the end of the GIT for treating inflammation. Similarly, for the anticoagulant drug also, we can employ from this polyepsilon, even this caprolactone has been employed. Now, apart from the immediate release and extended release formulations, electrospinning has been employed in the tissue engineering. Tissue engineering deals with the development of the various from the biological substitutes for replacing the diseased or the damaged tissue in the humans. Examples of tissues are we can employ tissue engineering for treating the defective skin or for the cartilage, heart, or the bones. Bone tissue engineering, here the electrospin nanofibers or the nanoparticles can be employed in the tissue engineering applications. So the various types from the commercial electrospin products which are available are the AOs, which is employed for wound healing. Zeus Company Inc. consists of the non-woven fibers for the sutures, rethink, surgical masks. It is specialized in the commercial scale manufacturing of the advanced from the biomedical electrospin nanofibers. Nano Dream, employed in case of the textile nano layer. Nano from the fiber clothing. Bria Safe for the air filtration. These electrospin masks have been employed for the filtration of the air to remove the toxic gas and the dust. In the tissue engineering, the electrospin nanofibers have been employed as scaffolding for the tissue from the regeneration growth, the wound healing, and electrospin nanofiber dressing, it can be employed. It allows and it will increase the oxygen exchange and will help in the wound healing process. Similarly, in the air filtration also, the electrospin nanostructures have been employed. And in the defense, this can be employed in the area of the warfare toxic gases. Electrospin nanofibers, they can remove the toxic warfare chemical stimulants. And electrospin nanofibers can also be employed as a face mask, swasa face mask, odorless plus. It can be also employed as a device that is in the Nanotech Private Limited, eSpin Nanotech Private Limited. Similarly, the swasa surgical mask, it can be employed as a device in the surgical face mask. PK from this Papyrus Biotronic, Berlin, Germany, electrospun polyurethane fibers on the stent. It can be employed thin, highly elastic from the membrane. Agiclot, sent Teresa from the Medical Inc., Egan, USA, Dextran electrospun nanofibers, fibrin sealant designed specifically for the bone bleeding. Surgical implants and the wound dressings, bone void, defect filling material, components are the beta tricalcium phosphate, bioabsorbable polymer, and the SIV, that is the silicone containing calcium carbonate that promotes the bone formation, Ribosys ortho rebirth, Heal Smart Poly Remedy Inc. for the anti-infective dressings with the addition of the HA, that is a hyaluronic, even this uronic acid, it can be employed for the wound healing. Commercial electrospun products for the healthcare are the beta tricalcium phosphate, siloxane containing calcium from this carbonate, BioWeb, ZS, USA, PK from the Papyrus, Ribosys, Surgiclot. Surgiclot can be employed as a wound dressing for the wound healing process. So the extracellular matrix protein found in the bone. So here the various ECM proteins have been employed, like from the collagen type 1, type 5, osteocalcin, osteopontin, bone, sialoprotein 2, osteonectin, fibronectin, thrombospondin. So all these type of extracellular matrix proteins have been employed in the tissue engineering studies. Various types of the polymers have been employed in the bone tissue engineering studies, like the synthetic polymers, polylactic acid, polyepsilon caprolactone, natural polymers like the chitosan or the silk from the fibroin, polymer blends like the polyepsilon caprolactone, gelatin, inorganic like the bioactive glass, hydroxyapatite, and even the silicate, composite or hybrids like the gelatin hydroxyapatite, and even the chitosan hydroxyapatite, siloxane from the gelatin, polyepsilon caprolactone, beta tricalcium phosphate. So here the various electrospun nanostructures have been prepared, and the various fiber diameters hang as low as 20 nm to as high as around 630 nm. So these have been employed to, in the case of the tissue engineering studies, these type of the electrospun nano from these fibers have been employed. So the composite or the hybridized nanofibers, chitosan plus polyepsilon caprolactone or the polylactic acid plus bone bioactive inorganics like hydroxyapatite, fluorohydroxyapatite, and the silica calcium phosphate, and the bioactive glasses have been employed. Gelatin plus polyepsilon caprolactone, polylactic acid, and the bone bioactive inorganics have been employed. Hydroxyapatite, fluorohydroxyapatite, and the silica calcium phosphate, bioactive glass or the bioactive glass from the ceramics have been employed to improve the bone extracellular matrix ECM. Gelatin plus polysilane has been employed along with the CaCl2. So here electrospinning is a membrane fabrication technique in which highly porous structures can be generated from a variety of the drugs and excipients. It is highly from the versatile technique in which the operating, here we have to optimize the electrospinning process. by altering the various from this, like what are the excipients we're employing, drugs we're employing, absolute from the viscosity of the excipients, surface tension, voltage range, flow rate, syringe to the drum distance, electrospin, nano from the structures can be obtained with a wide range of applications, the filtration, bio from this, even the engineering studies, textiles, electrical engineering, and the tissue engineering studies. Electrospin nanofiber scaffolds or the membranes have found numerous potential applications in almost every field, including the cosmetics, clothing, tissue engineering scaffolds, drug from the delivery applications and the wound healing applications. Electrospinning technique it plays a very important role in the various from the fields and electrospinning technique has exhibited excellent potential in the various fields, specifically in the tissue engineering applications. So these are the references. If you have any doubts, you can ask me. Thank you.
Thank you, Mohan Verma, sir, for delivering such a wonderful and insightful presentation. So this technical presentation will surely enlighten the minds of the young researchers, and more and more research will be continued in the field of electrospinning. So participants, if you have any questions, you can ask. Okay, sir. Let us move to the vote of thanks session, sir.
Okay, sir.
So,
Thank you.
good afternoon, everyone. On behalf of Sri Vishnu College of Pharmacy and the Department of Pharmaceutics and Department of Pharmaceutical Quality Assurance, I extend my sincere vote of thanks to all those who contributed to the success of the today's webinar. I express our heartfelt gratitude to our speaker, Mr. Ravi Kumar, and Dr. Mohan Verma for delivering such insightful and informative sessions. So, your valuable knowledge, practical insights, and engaging presentations have benefited all the participants. I would also like to thank our management and principals for their consistent encouragement and support in organizing the webinar. So my sincere appreciation for the organizing team, faculty members, technical support staff, and volunteers for their efforts in ensuring the smooth conduct of the program. So I also like to thank all the participants for their involvement and interaction in the session. So we hope this webinar has been informative and useful to everyone. Thank you once again for your time and presence. Have a great day. Thank you. So
Thank you, sir.
with this,
Thank you.
we are signing off this session, sir. Thank you.
Thank you, sir.